Okay. Good morning, good afternoon, dear friends, colleagues, viewers. Welcome to the channel Innovative Sustainability. We continue our series of presentations with different aspects of sustainability and innovative techniques that can help to solve several issues. Today we have Dr. Marc Taraban. He's joining us from US, from Rockville, Maryland. And he will be talking about the power of water, a very interesting technique of low field NMR. So just before he begins, a brief introduction. Marc has graduated from Novosibirsk State University. He received his PhD from the Siberian Division of Russian Academy of Sciences, specializing in biophysics and physical chemistry, I believe. And then he continued his journey as a visiting professor of University of Utah. Then he joined University of Maryland and he held several positions there. And he's currently associate research professor at the Department of Pharmaceutical Sciences. He's also part of the institute for the, I think Mark, you need to help me here.
Bioscience and biotechnology.
And biotechnology research. So thank you very much. Marc, the floor is yours. Welcome. Welcome to the channel. Thank you for joining today.
Yeah, thank you so much, Marina, for inviting me for the presentation. And then good afternoon, good morning to everyone. Let me share my screen.
Okay. Yes, it's coming up slowly. Yes, here you go. That's the one. Perfect.
Okay.
Yes.
So today we will be talking about the fascinating technique which we call the water proton nuclear magnetic resonance, and that's a non-invasive analytical technology which might be used in very different applications all over the different branches of human interest and human industry and manufacturing, including the pharmaceutical sciences. Basically, I will be concentrating on pharmaceutical sciences applications in the pharmaceutical domain, but we can also briefly discuss other applications of this technology.
That would be great, yeah.
To start with, I just want to remind the audience that water was the sample which have been used by Felix Bloch when he was discovering nuclear magnetic resonance spectroscopy, for which they will be getting the Nobel Prize in 1952 together with Edward Purcell. They have been using just a bottle of water as the sample, and you can see on the right side, on the top right side of the slide, the signal from water, which is just weak and bleak and barely visible, but it's the first signal of magnetic resonance that has been ever detected back then in 1946. Currently, water in most cases is considered to be a nuisance for measurements, because if you look at the left panel, you can see there that the signal of water around 5 ppm in high field NMR is strong, even with the water suppression technique. And the signal of the protein after 100 scans in water provides the very bad resolution, and you can barely identify different groups from this spectrum. As for the pure water signal on the right side, on the right panel, you can see that it's at the 5 ppm. After one scan, you can see nice and clear signal. This is the reason because the water has more than 90% concentration in the system, and you can see the signal clearly and nicely with the very high signal to noise ratio only from one scan of measurement. However, water is not a nuisance from our understanding. We are trying to explore the power of water signal, and water seems to be a very potential reporter of the events that happen inside your sample. Benchtop NMR instruments with the wide bore might be capable to accommodate drug products in their original containers and non-invasively analyzing by using water NMR. So, and the reporting parameter of that is the relaxation rate of relaxation of water, R2H2O, which is obtained from the decay of the intensity of the signal, and the detection is usually from seconds to minutes. Mm-hmm. So, slides.
Yeah, we're on slide number 4.
Okay. So why water is a reporter of all of these events? There are several mechanisms that are capable and provides the capability of water to detect the changes. For example, aggregation of the protein results in a large assemblies with the faster relaxing protons. And then during the proton exchange, water have faster relaxation rates, so it can sense the aggregation. For magnetic susceptibility contrast, for example, we have aluminum adjuvanted vaccine where the aluminum particles have a larger magnetic susceptibility compared to water. There will be a magnetic field gradient on the surface of the particle, which results in also an increase of relaxation rate. And in case of the clustering of nanoparticles, for example, or other objects like cells or other aggregates, the water inside the cavities starts to move slower compared to water outside. And during the diffusive exchange between these two water molecules, the result will be a faster relaxation of the bulk water too. So what is the capabilities of water proton NMR technique? It's a non-destructive inspection technology which uses the water molecule and drug products in a process flow as a signal source. Water is interacting, as we mentioned in the above slide, with all ingredients of drug product and reports information of their aggregation organization, and is a reporter of many different critical quality attributes. And most importantly, water NMR collects all of this quantitative data non-invasively and allow contact-free, in-time, real process monitoring, static and flow mode for continuous binary manufacturing. Here, I need to underscore again, we are not opening dark product containers. The analysis could be done. Yeah.
I just wanted to highlight that, think for viewers that this technique you're basically using without, destroying the material. It's basically you're measuring and you're saving material, and plus sterility is not compromised. So the same vial can be continued on, to be used for commercial purposes or for any other purposes. It's intact. So I just wanted to highlight because it's not.
It's not only the vials, it's also the injection pens like insulin pens or GLP injection products. It's also the prefilled syringes with the antibody injections, for example, glass. Even the stainless steel needle is not interfering with the measurements because stainless steel is non-magnetic material. So all of these drug products, after analysis, if they considered to be authentic or in a good quality, they can be used by the patients because, like in MRI measurements, you are not hurting the sample. In case of MRI, the sample is the patient. Here, the sample is the drug product in their original container. And this technique is using rather small and compact and inexpensive wide bore benchtop NMR instrument with the permanent magnet. So you don't really rely on electricity, for example, for cryogenic or electrical magnets. You don't need this permanent magnet is providing you with the required magnetic field.
I think this is also a good point to highlight, because normally NMR, like maintenance, keeping it requires special facility. It's a lot of work, not just to have skilled personnel who operates it, but also to have facilities like equipment and continuous need for nitrogen or helium, right? So this is something that heavy on the operational costs as well as the front costs as a capex. So this is different. This is what you just mentioned. The magnet is constant, so you can operate as is, as long as it works, right? And what would be the average lifetime for the instrument to use from your experience?
I mean, with the permanent magnet, I mean, it's the magnet will live forever. The problem might be with electronics, but it will live as any conventional electronics. Some instruments that we have here, for example, the one you can see on the left panel on this slides is made in 80s. So it's I don't know exact, probably more than my lifetime, I would expect. So here you can see a small slide, small photos of the family of different benchtop NMR instruments. You can see that you can put the whole vial, like for example, in the central panel, you see a 20 milliliter vial with the USP saline could be lowered into the magnet and measured there. It's possible to characterize different drug products non-invasively. Here you can see for example, the insulin pen, which is lowered into the instrument, the vaccine vial, which is lowered in the instrument. So all of this can be done non-invasively and pretty fast.
Vaccine pen on the very right? Look with the west. Okay, yeah, got it.
Yeah, on the very right, this is a vaccine in a sample holder. Yeah,
gotcha.
It's on the very right. And here is the flow NMR for the monitoring of the process flow directly, non-invasively. You can see this is the magnet here has the through hole, and you can put a different flow cells into that. You can put the pipe with the product. You can put the special cells with the coiled coil to increase the productivity of that instrument. And it allows you to monitor the process flow under different continuous monitoring conditions. Here, there's a photo of the facility where I'm working. This is the Institute for Bioscience and Biotechnology Research in Rockville, Maryland. This is the joint facility between the University of Maryland system and National Institute of Standards and Technologies. It allows the very intense collaboration between the standard setting authority of the United States and our university. We are in close contact with the scientists here, and we also often work together with regulatory scientists like the scientists from USP, United States Pharmacopoeia, and United States Food and Drug Administration, FDA. Here to show you, this is our facility, benchtop NMR suite in IBPR. There's in this pretty small room, we are hosting 4 different instruments. So 2 of these instruments are temperature controlled, and one of them is flow instrument, and one of this instrument has the robot, which allows you to position the sample inside the instrument at the most optimal position for the highest signal to noise measurements. And this is just to summarize what we have already demonstrated. We have demonstrated the applications of our technology in different domains of the pharmaceutical industry. In the formulation domain, we have demonstrated that our technique allows you to monitor stability and integrity of drug product and drug substance. In continuous manufacturing, you can monitor the critical process parameters like the flow rates, concentration of the solutes or proteins or whatever you need to detect in the process flow, aggregating conditions, and at the point of release and point of care, as we have already discussed a minute ago, you can measure the sample, and if the sample or drug product or the vial or the syringe or the insulin pen is of good quality, then you can give it back to patient. If it's not, you can discard it or give it to authorities for better detection of the reasons of that.
Characterization, yeah.
So and also we can do the different measurements. We have demonstrated the quantity uniformity, published a lot of papers on that for the different MAP formulations, aluminum adjuvanted vaccines. We have demonstrated the sensitivity of our method to detect the empty and full ratio and viral capsids for gene therapy products. We have shown that the aggregation, agglomeration, clustering also could be detected by this technique. We can also show that we can detect the cell counts and viability of the cells in cell therapy products. And most importantly, this is the detection of substandard and falsified vaccine and biotherapeutics, which is a hot topic right now for the fighting for the counterfeit and falsified products.
True. I think on counterfeit, I just looked up, it was, I think for 2025, the projection around 4.2 trillion US dollars of loss worldwide. And so, of course, they're talking about all sorts of products. But if we look at specifically, so apart from economic loss, they also mentioning that normally counterfeit is produced from low quality materials and they deteriorate faster. And so they become environmental concern because they end up in the landfill. But in terms of pharmaceuticals, since we're focusing on those, this is not just loss financial, it's also a hazard for human health. It could be a loss of health, it could be loss of life, depending on the severity of reaction and a potential harmful content of the counterfeit products. But also I know that every company in one way or the other looking into that. And I think from what I'm hearing in this presentation and from our collaboration that in the past, this is one of the techniques that we could call disruptive. It just gives you different look at the whole process of testing, measuring. It's with more concern about not to generate waste in the first place. It's to preserve a sample, but also it's to find answers much faster and reduce the number of tests that are normally done extensively for drug product vaccines, for that matter. It doesn't eliminate their characterization, but definitely when you know your process, you can really cut off to the most essential methods, such as this. So this is how I see it. Like it's it has potential to kind of, shift the balance towards like minimize the efforts on the testing and minimize the losses from all points of view.
Yeah, and if you look at this also from the viewpoint of the current weight loss drugs popularity, there is a huge number of reports of the counterfeit GLP-1 products. And there is a lot of online pharmacies who are providing this, and some of these pharmacies have no license, they are not certified. So buying this from them, you are risking to buy something which might be useless, at least, or dangerous at most. So I think that's a very critical problem right now and very hot topic. The analysis of authenticity and quality of GLP-1 weight loss products, and this technology can do this analysis without taking anything out. And if the product will be considered to be authentic, you can give it to patient. If not, then you can go to the full characterization mode and use the HPLC or mass spec or other technologies that allows you to characterize and find out the reason why this particular product, why this particular vial or dosage is bad or counterfeit or falsified. But in general, the total approach here, because here you can analyze every vial, every drug product container. Some of these technologies is mostly could be automated easily. And because of the measurements is very fast, it might be, you might down it to the several seconds, like 15 to 20 seconds of measurements.
For a while.
You can analyze with the high throughput system, with the robotic system, with the automated system. You can analyze a lot of drug products very fast and discard those that are considered to be counterfeit. Or, for example, you have some kind of the imported drug products batch, and you have no idea. Right now, all that people are doing, they look at the so to say outer look of the product container, whether it's the barcode there, whereas the label is correct, whether the label is not shifted. Like, for example, recently, as far as I know, FDA has been detected their batch of the counterfeit, non-authentic GLP products. I don't recall which was this exactly, but they were able to find it out only because the expiration date was on the other side of the pen, not on the side of the pen which is used to be. That's the only way. Now the counterfeiters know if you will put this on the right side of the pen, then that will be very difficult to figure out whether it's counterfeited.
Exactly.
Yeah, and that's the problem. I mean, it's a great problem, and I think that the use of this technology is very helpful and might save lives. Literally, might save money and might save environment by decreasing the waste number.
True. It's several issues. Exactly, it's health, life, and eliminate waste. But I think also, from both economic impact and it just also shows different view that with this technique you can, I don't know, potentially place it in different organizations. For example, customs, for example, where you need to take action, or testing labs that are working with government entities and required to get quick results to identify problem very quickly. I think that's basically, as I mentioned before, it appears to be a disruptive technology that can force us to look at the whole picture of efficiency in pharmaceuticals completely different way. Because I think cost and efficiency is a big issue for any industry, pharmaceutical included, but also beyond. And even in healthcare facilities, just additional tests for the peace of mind, considering that they sometimes have different vendors. How to check that, like you said, what do you have in a vial? Is it correct or not? By having libraries, I know you will have in features, the following slides, more details about it. But it's possible to identify whether it's correct or not, and also what comes in mind, maybe because also here in United Arab Emirates, the big emphasis is now on machine learning and artificial intelligence is to train the machine to quickly recognize whether it's on a mark, meaning it's a correct sample, it's what's supposed to be by a certain company produced, or it's different, and then quick action has to be taken for that particular batch. So, Mark, I think in terms of time, we'll probably stop here. So for the viewers, please stay tuned because we'll have a second video, and Dr. Marc Taraban will continue discussing, and also will show more details where the this technique is shown in action. And I think you will see that it directly contributing to sustainability because it answers many questions, environment, health, many aspects that currently sustainability is focusing on. Marc, thank you very much. Please stay tuned. We'll rejoin very quickly. Thank you.
Thank you.